All right, welcome everybody. We're going to give a couple of minutes here after the hour to get everybody on. Okay. Still got some numbers jumping up here, but we're going to get started in just a minute. Give people another minute or two to trickle in. All right. We're looking a little stable, so I'll start with just a couple of announcements. So good morning, afternoon, anywhere in between, depending on where you are. I am Eiley Allen. I'm the general manager of Wilderlab US, and I'm filling in for Sean today doing the intros. Just a couple of housekeeping items. We have our next June webinar scheduled as well, so keep an eye out for the date and time and the meeting invite for that. It'll be Di Gleason, so you won't want to miss that one either. And just as a reminder, this meeting and all of our webinars are recorded. So if you know anybody who would enjoy Shelley's talk after this, you can feel free to send them around. Again, that link will go out and it'll be hosted on our Vimeo channel. So that is just all the two announcements I have today. Keep an eye out for June and then, I'm sorry, May. I'm 1 month ahead of myself. It's only April now. So Di is our May seminar series. And I think we still have a couple of people trickling in, but I don't want to take too much of Shelley's time away from her talk. So I'll give her a quick intro. I'm excited to announce that Shelley McMurtrie, principal scientist and co-founder of EOS Ecology in New Zealand, is today's speaker. Shelley's going to tell us a little bit about eDNA Insights, which takes complex eDNA data and turns it into clear, analysis-ready information. So Shelley, I won't steal any more of your thunder. You can share your screen and pop on, and then we can have the Q&A is going, the chat is going. At the end, there'll be plenty of time for questions. So save them up for Shelley and her team.
Okay, are we seeing?
Looks fantastic.
Presentation, wonderful, that's working, love it when technology works. So kia ora koutou and ngā mihi Eiley for the lovely introduction. As Eiley has said, I'm here to talk about eDNA Insights, which is a service that provides an automated and science-supported interpretation of your eDNA lab results, giving you the power to quickly summarize and easily interrogate your eDNA data, minus all of those headaches. But before we jump into talking about data outputs, I thought it was probably worthwhile to just quickly dip our toes into the subject of eDNA. What is it? How does it get there? How is it collected? And what affects its detection? Environmental DNA, or eDNA, is genetic material that's shed by organisms in their environment. Skin, hair, scales, feathers, mucus, saliva, feces, urine, as well as being a bunch of random words. They're also all examples of sources of genetic material that can be shed and end up in our environment, which means that we can then go along and collect that shed DNA and test it. In Aotearoa, we can get eDNA testing done on soil samples and on water samples, but the focus of the talk today is on the water samples. When we think about the DNA that's in the water, this can come not only from those things that live in the water, but also from those that live on the land. DNA from organisms that never interact with a waterway can actually still end up in the water. So material that is getting blown around or that's on the land can get washed into a waterway where it can potentially get picked up in a water sample. So how do we go about collecting that eDNA sample from our awa? Typically, this is done by using either an active sampler, so that's a syringe that collects water and forces it through the sample filter that's shown on the top left-hand side, or by setting passive samplers overnight, where the water passively moves through the sample filter. And that's shown with the lovely Wilderlab shot that's at the bottom left of that slide. Both of those methods involve filtration. So that's where water is passed through a fine porous membrane, which traps the cellular and subcellular material. That sample is then preserved on site and then packed up and sent off to the lab, such as Wilderlab, for testing. But whether we're able to pick up an organism's DNA in our water sample depends on a bunch of factors. This includes things like, where are they found? Are they living in the water or living on the land? What's their biology? Do they secrete a lot? So do they shed a lot of DNA? What's their size? What's their movement? And so on. And also, what are the conditions like? Has it rained recently? What's the speed of the water? Its salinity, temperature, turbidity. So what we're actually collecting on our filter and what affects our ability to collect it in the first place. Once again, this is dependent on a number of things. The first is how DNA is produced. So production is the shedding of DNA into the environment, and that largely depends on the size and abundance of an organism and its biological and physiological features. Take, for example, fish. They can release large amounts of DNA into the environment compared to something like arthropods, which are crustaceans and insects, which release a lot less DNA. Production can also be influenced by metabolic rate and can even change throughout an organism's life cycle. Think about when fish are spawning. There's obviously a time when they're releasing a hell of a lot more DNA into the water. But an organism doesn't need to actually be alive to shed DNA. What that means is that the DNA we pick up in a water sample isn't necessarily only from things that are living in the water or on the land. It can also be from things that have been discarded in our environment. So, depending on where you're sampling, you could end up sampling someone else's discarded lunch or rubbish. The second factor is degradation. So that's how quickly DNA degrades, and that depends on physicochemical and biological factors, things like temperature, dissolved oxygen, pH, ions, even microbial activity. For example, studies have shown that macrobial eDNA, and that's just a fancy word for DNA from larger organisms, that can persist for longer in colder and more alkaline conditions. The consequence of degradation is a reduced amount of material that is available to be collected. But degradation can also lead to some chemical modifications of those DNA chains that are still hanging about, and that can hamper the correct identification of a species. And finally, we've got transportation, which is really just the passive movement of DNA in the environment, such as it being washed along by the flow of the water. That means that what you collect may actually have come from something living or something dead that was some distance away. And that also cycles back to that degradation issue, because if it's been in the water for longer, it may have become degraded. This is all something to keep in mind when we look at results from an eDNA sample, as your results aren't telling you what is found in the water at your site, just what was able to be detected in the sample of water that you collected. And we'll come back to this later on in the webinar. So we've gone through the process of collecting our sample and sending it off to our friendly eDNA lab. At the lab, all those tiny bits of DNA that were collected on the filter get extracted and amplified. So that effectively means making copies of the DNA. Then this is read as DNA sequences. Those sequences are then cross-referenced against a DNA database to work out what organism the DNA belongs to. Depending on the condition of the DNA, this may be enough to get an ID to a species level, or it may only get it to a broader level of classification, such as family or class. This information is then turned into a results data file, which tells you the taxa that were detected and the strength that DNA signal was. One of the great things that you get as part of your lab results file from Wilderlab is the TICI score, which stands for I think it's the Taxon Independent Community Index. The TICI score is a number which gives you an overall sense of the health of your site. But like every other biotic index out there, it doesn't tell you about the species that were detected and what that could mean. There's also a wheel of life that's sent out with your results. I love these wheels. They look great and they give you a sense of what groups most of your species are found within and where their connections lie. But other than a general overview, there's probably not too much more that can be pulled out of this. The key output from your eDNA lab is found in the Microsoft Excel file. Opening the file from your lab is super exciting, but then you get to look at the species list and you think, okay, cool, now what? There is a huge amount of really useful information in this file, but a name on its own doesn't tell you very much, and it's not a ready-to-use type of file from an end-user point of view. It's not linked to your site numbers. There's no other information about the detected species, such as their phylogeny, whether they're usually found in freshwater or whether they're a terrestrial or a marine species, you know, what their biostatus is, what their threat classification is, and so on. On top of that, for data analysis nuts like myself, it's not set up in a way that means that you can easily analyze the data. And that's okay, you know, because let's be fair about this. It's not really the lab's responsibility to make ecological sense of your data. Their expertise lies in the really tricky process of extracting the DNA and linking it with a massive library of DNA sequences. They shouldn't be expecting, we shouldn't be expecting them to also work out the ecological significance of the organisms found in our sample. Making sense of your data from an ecological standpoint, well, you know, that's best done by ecologists and data analysts. And that's where we come into the picture. As scientists, we spend a lot of our own time working through eDNA results, wrangling files into a format that we can pull out relevant information and run analyses on. We work with a lot of catchment groups and other practitioners, and we know that this is something that everyone's struggling with. So we decided to work out a better way of doing things, a way to create an automated process that interprets eDNA lab data files and creates meaningful outputs that tell you more about your data and save you lots of time and angst in the process. The process allows you to upload your data file from the lab, along with filling in a few easy questions, things such as your contact deets for the submitter and the data owner, that type of thing. The data file and this information then goes through the eDNA Insights process, and a Microsoft Excel file and a web-based report summary is produced. And these are then sent back to you as the submitter. Now, obviously, this is grossly simplifying the Herculean task that goes on in the Insights process itself. But the overall process is actually as simple as this, such that within minutes of putting a file through the system, the outputs are produced. So what do you get? As we've said, you get two files. The first is a summary report and the second is a Microsoft Excel file. Both of those files are sent to you via email. They're discrete files, which means that you can hold on to them yourself on your own computer or system without worrying about who's got your file. The report summary provides an at-a-glance overview of your data, looking at what was found across all sites in the lab data file. An interactive map shows you where you sampled, and then your results are summarised based on key biota groupings, things like algae, invertebrates, fish, birds, mammals, plants, and so on, as well as by the primary environment type that they are known to be found in. Each section can be expanded and collapsed as you work through the file. Section 7 to 13 then give you a further breakdown of some of the key taxa groupings, and it shows you and lists the number of species with a conservation and recreation status, biostatus is more. And you also get a nice species table listing of the key species for things like birds, fish, freshwater invertebrates, for the clean water taxa, mammals and plants. So this is a perfect way for you to get an immediate appreciation of what was found, and it's also great as a quick copy and paste of key overview findings that you can then use for your own report outputs. The Microsoft Excel output file will be of interest to all those data enthusiasts out there. This file holds a huge amount of information about each and every species recorded and from each and every sample. The first tab, which is tab A, this just holds the metadata about your data file and where it came from. The next two tabs, which are B and C, are just a presentation of the raw eDNA results as they've come from the eDNA lab file, with the addition of linking it to a site number, a sample date, and presented in two different ways. And we've done this so that you can always go back to the raw data that originally came from the lab, but it's just presented in a more usable format. However, it's really in the next three tabs where the magic happens. So tab D and F is where the full eDNA Insights process has been run. There is a mind-blowing amount of information in these tabs. It takes a while to sort of work your way through it. But the power of these outputs is that you can do whatever you want with your data without having to go looking for further information. Tab D and E hold essentially the same information, but it's just presented in two different ways, which is helpful for how you may want to do further analysis on your file. But for both tabs, they show each species recorded in each sample and link them to a hell of a lot of columns of related information, which tells you everything from their conservation status and biostatus to migratory classification, weed and invasive species flags, their phylogeny, and even if their detection level is questionable or not. On the other hand, tab F provides a summary of those outputs, and this is presented on a per site basis. So like your eDNA Insights report output, this provides a high-level view of your data, but unlike the report output, this is presented on a site-by-site basis. And finally, the important information tab contains more information about what each data tab contains, which is a super helpful guide when first viewing your Insights Excel file and to sort of get used to it. To look further into the value of that E tab, the way that the data is set up in that tab means that you can do pivots of the information in a few easy steps. This gives you the power to do what you want with the data without having to spend those hours to begin with to reorder the data and link it with those key biostatistics. If we focus just quickly on the bottom right-hand pivot example, this is showing how we deal with duplicate levels of ID. So this is an example whereby we've flagged the genus level where there is already a species within that genus that's been recorded at the site. So this allows you to quickly remove the unwanted duplicate IDs at the touch of a button. But what do we do to get these outputs? All of the work that goes into our data, so all of the work that we do kind of sits within that data processing section. And this process relies on the sound advice and insights from our ecologists and scientists, data specialists, and AI engineers. And I'll take you through some of the key steps and processes that we run. The first thing we do is to remove the extraneous dross from the lab file. The raw lab file includes results at all different levels of classification. And as we sort of talked about earlier, some of the reasons for that could be that the DNA strands in your sample may only be partial, so they may only be able to be linked to that broader level of classification. And it's totally appropriate for a lab to provide all that data to you as it's what was found in the sample. But it does create a lot of non-usable data, and it does add a level of repetition to the data file. And this is sort of shown in the example here for the chaffinch on the right-hand side, where you can see that it's recorded not only at the species level, but also effectively at the family, order, and class level. On top of that, we've got the issue that classifications such as biostatus, threat status, all of those things are only applied at the species level. So if we want to know what's really going on, we do need to focus at that species level. And honestly, that's where the power is with DNA files, as well as the fact that they are going down to that species level. So what we do is that we bring across the genus and the species level data, and then we indicate for each sample whether you can remove the higher genus level specification if that species, if a species within that genus has also been identified. And that was shown in the previous slide. So removal of the broader taxonomic classifications allows us to focus on what counts. We also remove some taxonomic groups from the eDNA insights process. And that's just because there's currently a lot less known about them. And so they're not really helping us to interpret what's going on at a site. Now, this can change as we find out more about these taxa groups, but for the moment, removing them certainly helps to sort of cut out the noise from your data file. Should you want to look into these more, for example, if you're a specialist in bacteria or fungi, you can still find them in the tab B and C of the Insights output file, and they're also listed in the excluded tab, so you know exactly what has been pulled out. Another key thing we do as part of the process is to look at whether the eDNA results for each species found can be considered a true detection or not. And we do this by comparing each species detected at each site against 3 uncertainty type tests. The first, which is type 1, relates to signal strength and follows the rules that were developed by NIWA in their 2023 publication on eDNA. The second, which is type 2, looks at whether the sample collection type is matching the key environment that a species is known from. This means that if a purely marine fish is found in a river sample, then it's flagged as uncertain. And that can help with picking up things like sample contamination or you've just sampled someone's lunch type thing. And the final one, type 3, looks at whether the species is known to be present in New Zealand. And we do that based on information from NZOR and MPI. And if not, then it's flagged. What this does is it gives you insights into the strength of your data and whether there are some results that need to be looked into a little bit more, or whether you should be picking up the phone to MPI to let them know that we may have a new species record in the country. This type of information is provided at the site level in the Excel data output, as well as being provided in the report output, remembering that the report output is looking across all samples and sites. The reason why these questions around detection come up is because of the nature of eDNA, how it gets into our waterways, how it's sampled and so forth. A lot of what we touched at the start of this webinar is what this is about. Knowing more about your samples and what may not be a true detection, or at least what should be looked at more closely, really is a critical part of eDNA reporting. And the eDNA insights process, well, it does all that heavy lifting for you in regards to that. But the biggest challenge for us to solve was about how to connect the taxa names in the lab file with, well, with everything else. Because a name on its own, as we know, doesn't tell you very much. Is this thing native or introduced? Is it threatened? Is it a pest, a notifiable species? Should I be calling MPI? What environment does it typically live in? There's a myriad of questions that we can ask about that species. To answer them, we've developed our Master Taxa List, which is built to automatically match up a species name in your lab file with everything you could want to know about it. Without it, detection's just a name. With it, that name carries conservation status, New Zealand origin, pest and weed risk, regulatory significance, habitat, context, all of those things in minutes. But honestly, doing that just was not easy. There's plenty of sites out there that have this kind of information, but the challenge in all of this is that not one place holds all of that information. Different databases hold different pieces of the puzzle, and they all say it in slightly different ways. So the question for us became, how do you accurately and consistently match a taxon name from an eDNA results file with all of these disparate sources at once? When you then add in the challenge of working out what environment they're found in, well, yeah, okay, this is where things get even murkier. Different sources use different terminology, and every source classifies environments in different ways. You can take, for example, our freshwater migratory fish species. Now, they're typically considered by ecologists to be freshwater species, right? But because they spend part of their life cycle at sea, some online sources actually list them as purely marine species. So this is a wonderful bird's nest of complexity that our process had to work through. The challenge we faced was to take around 40,000 species names and match them up with the same species names listed on online databases that have the other information we want to link to. But, and it is a huge but, there is no unified taxonomy across all these different databases. A species name is not written the same in one site to the next, and no two sites will list the information in the same way. We solved this through a hierarchy of steps. The first was name standardization. Every name is resolved to a single canonical scientific name, the accepted official form, you could say. That canonical name becomes the universal join key for everything else that follows. Second is the backbone taxonomy, placing each species into the tree of life using 3 overlapping authorities. GBIF for global coverage, worms for mainly marine taxa, although they do cover a lot of other taxa as well, and New Zealand Organism Register, NZOR, for New Zealand specific organisms. These don't always agree, so curation runs in parallel throughout, flagging the conflicts and keeping an audit trail. Third is the species names are matched across all databases using that canonical form in full species lineage, which is what makes the matching reliable, even when source databases use slightly different naming conventions. And finally is the domain enrichment. This is where the list gains its power. Pest biosecurity flags, New Zealand origin and conservation status, migratory status for fish, EPT status for freshwater invertebrates, ecological traits and key environments, recreational value, game status, blah, blah, blah. All of these are pulled from authoritative sources automatically. The challenge, though, isn't just scale, it's consistency. As I've said, I think I've probably said it several times now, species names appear differently across every database we work with. Without resolving those inconsistencies first, any downstream join will fail. You'll just get nulls where there should be data. So our solution starts at the foundation of backbone taxonomy. Before we touch any enrichment source, every name is resolved to its canonical form and placed within its full phylogenetic hierarchy. And what makes this work is that it's automated. There's no manual lookups, there's no spreadsheet wrangling, there's no human bottleneck. When a new source is added or a database is updated, the pipeline reruns and the Master Taxa List reflects it. Every enrichment step, however, does run through continuous curation and QA. So if we want to know where a pest flag came from, we can see the source and the date it was last checked. Building the list is one challenge. Keeping it current, that's another challenge. We connect to a lot of external databases, but they don't all move at the same speed. So we've got to manage those updates through a tiered approach. Some sources, they're essentially static, right? So we check these on a lower frequency. A second tier updates every few years through a formal review. So the New Zealand Threat Classification System, that's a good example. That system gets updated roughly every 5 years. When those updates are published, we can rerun our process merge with those new classifications. So whilst these databases are, semi-infrequently updated, the changes are impactful and our process allows us to keep on top of that. The third tier is for sources that update continuously with ongoing revisions to taxonomy records, synonyms, and biosecurity status, those types of things. It is critical that we continually check these sites to ensure our system is up to date, and our automated rather than manual process ensures this happens. We also keep our taxa list up to date by flagging any taxa that are unmatched each time we run the eDNA Insights process. We can run periodic reviews of those gaps and formally onboard new taxa as eDNA methods improve or as new species are picked up through those online systems. The power of our setup is that all of the data we collate is primarily managed through our Master Taxa List. This gives us greater flexibility in managing our system, and it means that there's less work to onboard a new eDNA lab to the process. So at this stage, we've developed the end-to-end process for the Wilderlab eDNA results files based on their sort of output file layout from about late 2025. But there is no reason why we can't develop the process to also run using the Wilderlab data files. Basically developing a new data processing module that connects our Master Taxa List with the slightly different setup found in the Wilderlab's output files. So finally, probably the question that's been on everybody's mind, why we're not just asking AI to do all of this and save a whole bunch of bother. And it's a totally fair question. And the short answer to that is trust and the consequences of getting it wrong. AI large language models, they generate text by predicting what sounds right, not by actually looking things up. And they can hallucinate, producing plausible sounding information that is simply wrong. And in our field, that's really dangerous. Then there's the reproducibility. Ask a large language model the same question twice and you get a different answer. Different model version, different prompt wording, different type of day. In contrast, our pipeline produces identical outputs from identical inputs every time. AI is also a bit of a black box approach. You get an answer, but you can't easily fact check it. In comparison, every step in our process follows a series of rules, so we can point to exactly where it came from and when it was last verified. But, I'm going to be totally honest here and say we still use AI in our process. We use it to assist primarily with searching for any mention of key environment types that species are found with, because that is a huge task to do manually. So it is a bit ironic, given what I've just said, to say that we do use AI in a portion of our workflow, but it actually illustrates the distinction well. AI assists our workflow. It doesn't generate the underlying data. The pipeline is deterministic code. The enrichment comes from authoritative sources. AI is a tool we use carefully and strategically to produce weighted, curated outputs, but not to replace the rigor underneath. And that's the kitty of tools that sits behind our eDNA Insights process and what you get out of it as an end user. We know the time it takes and the headaches it creates to make ecological sense of eDNA lab data files. Working with catchment groups and as science practitioners, we know the value of providing outputs at the overview level, as well as getting down into the detail. So we've worked really hard to create a process that takes that pain away, to give you ecological insights that you can trust while still giving you the power to analyze your data as you want. So that's brought us to the end of our eDNA Insights journey. Not sure if I'm ahead of time or behind time, but if you'd like to find out more about any of this, just take a shot of the slide and you're welcome to reach out to me afterwards. In the meantime, I'll hand back to you, Eiley, and see if we've got time for some questions.
Perfect. I was just going to say perfect timing. No, not ahead, not behind. And thank you so much, Shelley. That was great. I had written down a few questions and it was funny because every time I wrote a question, 30 seconds later you answered it. So I do have a little list, but we also have some in the chat. Feel free to keep putting them in the chat or the Q&A. I will start in I can kind of synthesize. There's a lot around the geography. So is this specific for just New Zealand? And I think that was answered a little bit later in your talk, but one of the questions is, I think if you're looking to expand to additional geographies like Australia, perhaps.
Yeah, it's a good question, and it's actually lovely to kind of therefore see that there's people from outside of Aotearoa that have joined this webinar. At the moment, this is focused on Aotearoa New Zealand only. We haven't looked outside of that. Even though we sort of access with some of those international databases, we are primarily focusing on Aotearoa New Zealand for our first run at this.
Well, it does look like there's some interest, so exciting times for you. The other kind of group of questions was around the privacy, and so if you do have private data, just can you speak a little bit about how that stays if you load it in, then those online reports and everything, just making sure that it does stay private.
Yeah, look, it does. The way that the process will run, there's a number of steps where we analyze, filter, and organize the data and generate those reports. But when you submit your information, so that basic submission form that you have to fill out, who the data owner is, things like that, that passes through servers in Australia as a very sort of quick sort of process. But absolutely everything that we do in terms of the insights process itself, that runs locally. Your original lab data files, they sit on our local servers on site at EOS, and the actual eDNA insights process itself, that runs entirely on our local machines. And after the process is complete, we then email you your results. You can choose to do what you want with them from that point on, but they're not stored anywhere externally, and we'll hold on to those for a certain amount of time, and then we'll delete them from our servers as well. So we wanted to run, because of the factors relating to data ownership and concerns around security, we decided to take that sort of more stringent approach to begin with. So there's nothing that's sitting up on a website or anything like that at this point in time.
Awesome. Cool. There was also a question around if you can upload multiple data sets, and I think, again, you touched on that as well, and potentially different formats, but just maybe expand on that just a bit.
Yeah, so at the moment, the process runs through one file at a time. So the submission form relates to the file, the single file that you're submitting, but because the process runs so quickly, you just do, do a new submission form for each result file. One of the reasons why we do that is so that it's really easy to manage that process for you as the end user. You've got 5 separate files from Wilderlab for 5 separate lots of results. Each of those get processed separately, so that you've got your original file and you've got your eDNA insights outputs that sit with that single file. So it's just a cleaner way of running that process. I don't know if there was a second part to that question. I've forgotten. You'll have to ask it again.
It was just around the formats, but you did answer it in the webinar that you can be a little bit format agnostic as long as you're getting those key things, which was the species name and then the metadata, because then you sort of do the recalibrating to get you there, that your key species name that then plugs into the next section. So
exactly.
That,
yeah, that's right.
Yeah.
When we started this process, we kind of had a long conversation of how do you make a system more bulletproof, and so setting up that Master Taxa List allows it to be sort of agnostic. You've still got development time to read the file, that's coming from the specific lab, because every lab will have a different way of showing their results. But the iceberg, the base of the iceberg where all the work sits is actually in that Master Taxa List. So as a new output file comes along, the work to sort of create that matchup is less. And if, for example, Wilderlab update their file output, God forbid, it's not going to take as long for us to get that process up and running again.
Perfect. Great. We have 2 questions actually around cost. And so if you can talk about the cost, whether it's standard or if it varies, and access to the tool.
Yeah, look, it's a good question. It's been a bloody tough one for us, and we're still working through what a sample and potentially even if there's a batch pricing option as well. In our experience, and this actually is something I'd love to get some feedback from the folks that are on this webinar in terms of how long did they spend on wrangling these lab files into a format that they can use and sort of connecting up all those types of things. For us and for people that we've talked to, it can take between 4 to sort of half a day to 2 days to get the data file into an analysis ready sort of state. So, we're talking hundreds to thousands of dollars in terms of commercial rates for that amount of time. So it's obviously not going to cost anywhere near that much for a file to be run through this process. We think it'll likely be in the low to mid hundreds of dollars per sort of output file. So that's a very small portion of what you would otherwise be spending on that. But please, if people have got, one of the big things for us is to understand, what would people sort of pay for that, how much time, what is the value of saving you losing your hair, tearing your hair out over a data file.
Awesome. Thank you. We have another kind of cluster of questions around GBIF and reference databases. So how do you handle the differences between NZOR and GBIF New Zealand, or a more recent understanding of speciation? And then can you also get I think you could probably answer both of these. Can you also supply the full reference for true versus tentative detection rules, like, for example, NIWA 2023?
Yes, we absolutely can. I'm actually Zoe, if you're there, would you be able to sort of post that into the
somebody's actually linked it in the chat for us already.
Okay, awesome, brilliant.
The report's Environmental eDNA Guidelines and Field Protocols for Loadic Systems by NIWA. So obviously a New Zealand focused guidance document, but yes, somebody's put the link in the meeting chat.
That's fantastic. Okay, so that answers that one. What was the other part to that question?
Differences in reference databases and speciation.
Yeah, so that was the big one. So that's where we sort of run through and establish the tree of life for each species that comes through from the eDNA list. And that's been where the bulk of our work has been done, because how do you resolve the differences, even to the point that you can have a TICI SP with a dot and SP with the outer dot. You can get spaces, you can get ever so slightly different. A lot of the sources list the person who first described it as part of the species name. So that process all runs through to kind of go to work through, is this name the same as this name? Do we have confidence that these two things are the same? And if we do have that confidence, then it's matched to our canonical name for that taxa. It's not going to cover off absolutely everything, but we're getting an increasing percentage of success on that process. What we don't do is include the subspecies. And again, the reason why we've done that, if you go down below the sort of species level, is that the threat classifications and or the biostatus and stuff typically isn't connected to that. So there is some things that we haven't kind of included just because of that process that we're running through. We're trying to get the bulk, we're trying to sit at that happy median and at that species level or the genus level, which we will pull through if there's no species that's been identified. But even at the genus level, you've got limited information that's available in terms of its various biostatistics and stuff like that. So again, straight species is where the key focus is.
Awesome. And I really liked how you had the different kind of time frames of what you update when. when you make some of those updates, so if there's new entries on either a list or the databases, do you automatically rerun, or can clients request and then say, hey, can you rerun it ever so often when you do those updates to those kind of core parts of the pipeline?
Yeah, we haven't kind of worked that out yet. At the moment, we're thinking, this is a one-hit wonder in the sense that at the time and point that you've got your file, we run it through the system and that's what it is. And that kind of relates to the relevance, right? Because at that point in time, that is what the relevant information is. That's what we classified that species as, as a science society. That species was named this, on today, the 17th of April, 2026. So that is relevant to the time that file was processed through. The question of, running through stuff, okay, you want to historically go back to files and rerun it. Well, you can. If you've still got your original lab output file, you can just resubmit that and run it through again. And I know that, a bunch of us work on big long-term data sets where you're wanting to kind of drag the old data,
Definitely.
and compare it to the new. So at the moment, we're trying to keep this simple. So you would just resubmit that file and sort of run the process. But we're not, because we're not holding on to people's data, right. We're not dumping this into a big centralized database that's, feeding a monster. These are discrete units of information that we package up, we process, and we send back. And then for us, we remove that after a number of days, so that it's sort of like the submitter and the data owners, it's their stuff, right? So they would, because of that process, they would need to resubmit. Anyway, yeah.
Awesome. There's a question around terrestrial land species, and I think there was most of the talk had kind of a focus on aquatic, but can you do the same? Will all the same pipelines work through eDNA Insights with soil samples if you were to feed data from soil samples in?
That would depend on how those samples are. In theory, yes. Like at the moment, the processes on the taxa list, species lists that are dominated by freshwater species or species that get picked up in water. We do run this through for terrestrial species. So if you've got birds and mammals, terrestrial plants, terrestrial invertebrates, that are getting bats and things like that, if they are getting picked up in your eDNA water sample, they are processed through that process. So a lot of the stuff that we have in there, we've sort of got the dominant environment type as either freshwater or wetland, marine or estuary or terrestrial. So those things are in there. I haven't seen, we haven't looked into the format of the soil type samples, and obviously the taxa are going to be slightly different. So yeah, it would be interesting to sort of run that. But certainly this is not we only report on the freshwater stuff in your water sample. We're giving you everything that's there, terrestrial, the works.
Awesome. You're getting, we'll save the chat here because you're getting a lot of people reporting how long it takes them to wrangle, and there's definitely lots of interest in kind of having it be expedited. And again, another plug for us expanding to Australia as well. So
okay, good one. Thank you, people. That's exciting. Cool.
Yeah. I think I covered every question in the chat. If I didn't, just pop it back in again, or got to every question in the chat. I had one other question, which is kind of around presence absence. It seems like most of it is focused on presence absence, and whether you are thinking in the future of kind of expanding to quantity as well, whether that's quantitative PCR or using sort of inferences from number of reads after that filtering threshold. If you're kind of interested in anything more quantitative or keeping presence absence data feeding in for the type of reports you're generating.
Yeah, I mean, at the moment, we're just reading what's in the files, right? So we pull across the sequence counts, and we depending on how, because the different tabs set the data up in different ways. So when we're looking across the site, you've got the min, max read, you've got the median and the mean for that particular site across the reps if you're doing more than one rep for that site. But we're not the lab people here, right? We're not Wilderlab. We don't run the tests, the PCR tests and stuff like that. So we're literally just consuming the information that's in that file. This process has been like pretty mind-blowing and totally fascinating to work on. And I think we have delved into getting a greater understanding of how that eDNA sort of process works. But at the moment, it's just taking the sequence counts as they come through in that file.
Awesome. Great. I actually had a question around AI, which was one of the ones that you, as soon as I thought of the question, you did. But I think that's a great balance of kind of using it to speed up your processes, but not relying on it to do everything and make all of your inferences from there. So that was definitely of interest to me, and I'm sure there'll be more you can integrate to help with the speed up. We do have a couple more questions trickling in we have a couple more minutes left. So any other future plans for different insights or visualizations to come out along with the the web format and the Excel spreadsheet?
Yeah, look, absolutely. Like, for those of the people online that know us as a company, we are very passionate about science communication and effectively communicating complex data. So that is an absolute wish to be able to do some amazing visualizations. At this point in time, we've stuck with the nuts and bolts of, hey, let's get the system working and let's know that we've got trust and confidence in the outputs that we're providing. And so we're sort of grassroots in relation to that. Well, we all know that Excel files never look nice.
Those look like really great Excel files, I would say. A couple more nuts and bolts questions. Does the insight report come with explanatory notes with things like methodology or the date of analysis being run, just to keep track of all that metadata as well when it pulls through?
Yeah, it does. So I didn't click on it because it was a very long introduction section, but in the HTML report, the introduction section has the date that the sample was collected, all the metadata that comes through that submission form, as well as the date that the process was run. There's a references at the bottom that has some of the key sort of sources of information that we use, and then there is but most of that detail sits in the actual Excel file in the important information, where we have a description of each of the column headings and each of the tabs in terms of what that means and where that's come from. Yeah.
Awesome. And then a couple of questions on how to keep on when the tool become available, what the time frame for people to actually use eDNA Insights.
Oh my God, I wish it was like months ago.
Not to put you on the spot, but people are ready for it.
Look, I think it's probably, there's just a few more checks that we want to go through. I'm really happy with where it is, but, for us to just polish a few of those final outputs and stuff like that, I'd say, what are we now? I don't even know what month we are. April,
It is April. It is not May. Like I said at the beginning, I'm 1 month ahead, but it is April, mid-April.
So yeah, I'd like to think that in May we'll have this ready to go. The biggest thing for us is is actually setting up the system that people can, just email through to an email, and that pulls the information through, provides a link for the submission form and all of that. So we've kind of got the system running. Now we just have to do the add-ons to allow that to be a process that people can link into. And then, of course, how do we get paid? That's obviously really important. Otherwise, none of us have got a job. So also walking through, how we do that in the easiest way for people as well. Yeah.
Awesome. And again, that QR code, we'll post this recording on our channel, and people can get your contact info from there as well. Great. That was, amazing number of questions, Shelley. That's a sign of a great seminar. So thank you. You did an awesome job answering questions. I think the last couple ones have trickled in here, but again, really, really thanks everybody for participating and giving all the feedback on. I think you'll find it interesting to see how long people will kind of report for them to make their taxa list and or wrangle that data coming from the lab. lots of interest on when they're going to be able to get it out and use it. So as soon as you know, let us know, and we can send that out to our networks as well. And again, it is our May webinar. Di Gleason will be here. Look out for an announcement from Kendra coming up soon. And that will be a wrap for today. So just join me again in thanking Shelley. All right. Bye, everybody.
Hi, Kuna.